saw each other at I think I went to the IEEE meeting when it was in Berlin in 2019. So
back in the ancient times.
pre-COVID times.
Yep.
Yes.
Yeah. Okay, well, people are starting to join. I have a few announcements to make. So first, welcome to everyone to our February neuroprosthesis seminar. Baloo will introduce our speaker in just a minute. A few notes. Our Behind the Science series, which is on Thursdays. We have several of these scheduled. We have one for February. It's on the 16th. And that's concept reviews. That's next week. So if you have a project that you're in the sort of the conceptual stage and you want to get some feedback from your friendly colleagues in the FES Center, please let Cheryl know. We're trying to fill that up. Short, no preparation necessary, really. You just say what it is and we tell you what we think, okay? On March 16th, we have a speaker who's going to talk about what the media wants to know. So this is sort of some guidance on how to handle media inquiries into your research. On the 20th of April, we will have the topic is, well, it's right there, device design, Mark Cartolone, who's going to talk about the device design. And we also have First Tuesdays, which just popped up here. This is more of a science, internal science discussions, and Andrew Pieper, who is does many things, but he does one of the things of interest to us is he does drug development for traumatic brain injury, and he's going to be speaking on March the 2nd. And we have some other neuroprosthesis seminars that we're in the process of scheduling, but the next one that's scheduled right now is May 20th, and that's Monica Perez, who is now in Chicago at the Shirley Ryan Ability Lab and I guess Northwestern, definitely the VA, Hines VA. Okay. One other thing, and I thought that Andy just told me Andy Cornwell stuck on a call. You should have received an email from Andy about a new center that we have called VA-Team. And the T in team stands for translational, and I don't remember the rest. I don't want to, but this is a
education
VA,
and mentoring.
Education and mentoring. Okay, so this is a program that Andy and I and Robert Bonomo from the VA are leading. And it is to educate VA investigators on how to get their discoveries translated into the clinical world ultimately. And this, we just got the notice of award, and we started January 1st, by the way, but we just got the notice of award, and there's a deadline for applying to this program. So this program is educational, but you also, you go through some didactic materials, but you also are assigned a mentor and a program person to help guide you through how to go forward with your idea. I think it's a really good thing. You might have the best chance of getting in this time because we started advertising it like a week ago, maybe, because we didn't have the money till then. And the first proposals are due really soon. Do you know when, Erica, by chance?
February 6th. I don't know the exact date.
Might be
I
next
can look
week.
it up and let you know. It's not far off from a week from now.
We had to do it that way because of the way the program is set up. It depends on some other activities that are already scheduled. So we're going to be hustling the first time, but I encourage anyone who is interested in learning how to translate their Veterans Affairs technologies to participate in this. And you should have received an email from Andy about this. Okay, I'll stop there and turn it over to Baloo.
Good afternoon, everybody. It's my esteemed pleasure to present our neuroprosthesis seminar speaker and a personal friend of mine, Dr. Surjo Soekadar. So Surjo received his doctorate in 2005 from the Central Institute of Mental Health in Mannheim. And he was actually a visiting fellow at NIH, NINDS specifically, in the late 2000s, so about 2008, 2011, specifically at the Human Cortical Physiology and Stroke Neurohabilitation section. From there, he actually went to the University of Tübingen, where he was the head of applied neurotechnology, and the head of applied neurotechnology working group. And now he's at Berlin, where he's the Einstein Professor of Clinical Neurotechnology at the Charité University of Medicine, Berlin. He does really amazing work on merging BMIs and clinical translation for stroke and other disorders. He's also a great personal tour guide of Berlin. If you ever get a chance to go to Berlin, we had a great time. So I look forward to your talk, Surjo. And with that, I'll turn the floor over to you.
Well, thanks so much, Bolu, for the invitation. It's really a great pleasure to be here. And special greetings to Leo Cohen. I've seen that he's also here online. So hi. I'm going to share my screen. I've prepared some slides to share with you. And well, in my talk, I'm going to talk about the state in Berlin. Mainly, I'm also working a lot in brain stimulation, combining BCI with brain stimulation, but this will not be the topic today. But I will specifically ask whether BCIs are ready for clinical neurorehabilitation and broader adoption. So, well, first of all, of course, I have to give a little introduction to the BCI field, because there are also some graduate students who might not be so familiar with also the different tools that we are using now for brain-computer interfacing. So we can record brain activity invasively or non-invasively, and you're all probably familiar with MEG, EEG, but there's also some new technology now around that is based on quantum sensors, optically pumped magnetometers. You see this here on the upper panel. So these magnetometers can be placed directly on the skull, and as they don't need to be helium cooled, you can actually get much higher spatial resolution with these arrays. And we are now working at the PTB here in Berlin to build some high density OPM-based BCI to control single fingers. So theoretically, you can achieve a spatial resolution that is comparable to ECOG grids, but that of course has not been shown yet. But theoretically, because these sensors are much closer to the skull, and also you can do the source reconstruction because you're measuring neuromagnetic fields, you get a much higher spatial resolution compared to EEG. Of course, you can also use functional near-infrared spectroscopy that has been done for many years now, or functional magnetic resonance imaging. But obviously, these tools cannot be really used outside a laboratory. But it's a very important scientific tool to understand the mechanisms of, for example, neurofeedback paradigms. You are the experts for the invasive or implantable dimensions. So ECOG, of course, is one of the big pillars, and also these epidural grids that have been now developed in France by Louis Benabit. And lately, of course, we all know about the Neuralink technology that is using flexible threads. So we'll probably hear much more about this in the next, I would say, few months. And I know that maybe the video will be a bit interrupted. I try to improve it, but I hope you can actually follow it. So this is this link, the new device that has been developed with 1,000 electrode contacts that is implanted by this robotic device, evading blood vessels. So it promises to be less risky than implanting the Utah array. And of course, there has been shown that it has been used in pigs. You've probably heard about it. And they showed some signals, but of course, we don't really know how well these signals are really in reality. But we will probably see something. So I've heard some rumors that this device has been implanted in a monkey playing Pong. So maybe this will be published soon. Let's come back to the history of BCI for a moment. So the first clinically relevant BCI was introduced by Niels Bierbaumer, who's shown here on the upper right, who has introduced the BCI for locked-in patients in the late 90s. And he has shown that the patient who is unable to speak or move can modulate his EEG activity, slow cortical potentials, to select single letters on a screen and write a letter. So this has really started a huge wave of enthusiasm for restoration of communication with the BCI. And then the other big pillar in BCI research is, of course, restoration of movement. And you are one of the most important centers for this, I would say. So this is not new for you, but out of completeness, I wanted to mention this here. And now show what we have been working in Berlin. So I must say I'm not really working on implantable devices. I'm mainly working on non-invasive technology. So here you see the benefits of non-invasive. So first of all, if you implant, that's obvious. You have the risk of bleedings and infections. And right now there's still no device that has a certification for permanent use. And very often these prosthetic devices cannot be really used independently by the patients themselves. So usually there is someone around them and make sure that nothing happens. So now Myomo has a device that has been certified. So this is now being used as a prosthetic device. But essentially, if you have an implanted device, you want to have more degrees of freedom. And right now, there's no device that is certified for daily use that can actually provide this. So hopefully in the future. Of course, if you remove the devices, that's also another surgery and also entails some risks. So in Berlin, or at that time in Tübingen, I've been working on non-invasive devices and strategies. And here you see a number of patients who had a high cervical spinal cord injury. And you can see they have a hard time to grasp any of these different objects of daily life. So, for example, a book, a piece of paper, or a cup. So they don't have any strength in the fingers. And we equipped these patients with an exoskeleton that was implemented in a wheelchair. And with this exoskeleton that was controlled by EEG and electrooculography, they could securely grasp these different objects. And they could also leave the lab and eat, for example, in a restaurant. And the closing of the fingers was controlled by EEG when they thought about closing the fingers, and hand opening was controlled by eye movements to make it really safe for everyday life. And we've asked the patients what they would like to do. And here you see a patient who asked for some potato chips that he hasn't eaten for many years. And you can see how much joy he had with eating with his potato chips after so many years. So it can really improve quality of life. And control was very, very intuitive for those patients. Now we have also implemented this control paradigm in a whole arm exoskeleton control. Here we used 3D object recognition. So the object, for example, the bottle, would be recognized by the system, and the robotic arm would just be automatically driven towards the bottle and grasp it. So you would use EEG to initiate these different sequences of grasping movements. And yeah, well, these patients could also go into a restaurant and drink. Now, when we use the system on a daily basis in stroke patients who had no finger movements left, so no functional finger movements, sometimes for many years, we have seen something very surprisingly. So these patients use this neural exoskeleton for a whole month on a daily basis for 40 minutes. And after 1 month, we could see that some of these patients could move their fingers again. And when we looked into their bold responses during the attempt to move the paralyzed fingers, we could see that while there was bilateral activation before the BCI training, after 1 month of training, this activation reorganized and relateralized to the perilesional areas. And this correlated with the clinical improvement in finger movements. So this really shows that connecting the brain to a machine and having this operant conditioning can induce neuroplasticity that can trigger neuroplasticity, neurorestoration of movement. And here you see that when you do this, this is a work from some other group in Japan. They've also reproduced this finding, and they've shown that there is reoccurrence of EMG activity after approximately 3 to 4 weeks of training. Now, the same principle has been also shown for the lower limbs. So here you see a young patient who is driving this exoskeleton for restoration of gait movements. And here you see after 9 months, so this patient hasn't really received daily training. This was like once or twice a week. But after 9 or 13 months of training, he regained some function in his lower limbs and feeling, and also some basic locomotion. I hope it's you can see the videos. Okay. So now something that has been also very impressive in the last years is this work here from Nikolaus Wenger, who is also here at the Charité now. He has cut the spinal cord of rats, and these are the rats, and you can see that these rats can actually move. So he connects their motor cortex to a spinal cord stimulator, and with this stimulation, the rats could restore their ability to move. And the same principle has then been implemented in the work by Wagner in Lausanne. So they implanted these electrodes underneath the lesion. And here they didn't use cortical activity. They just used the EMG activity of the trunk, giving the instruction to move forward. And if they do this, the stimulation is turned on. And the stimulation is turned off, you can see that the patient is unable to move at all. Now with the stimulation on and the instruction to lean forward, move, the patient could walk. And they did essentially the same thing that we did with the stroke patients. And after 5 months, you can see that the patient could move his lower limbs. And after a few more months, you can see that this patient could walk unassisted, just using EMG triggered spinal cord stimulation. Now this shows the vast potential of neuroplasticity in these patients. And now in a recent meta-analysis, it has been found that using BCI in subacute to chronic stroke can yield very, very good results in the Fugl-Meyer. So actually it's in the higher rank of these different interventions that you see here. But of course, you can also see that the heterogeneity is very high of those patients who underwent the BCI training. And it also shows that this idea of putting a lot of like technology to the patients doesn't necessarily really improve. So it's really important to understand the mechanisms. So when they combine BCI, TDCS, and robotics, this didn't really show that this is more effective. So it means that we need to understand the underlying mechanisms much better, and then stratify and personalize this kind of technology to the individual case. So this is a suggestion here from Friedhelm Hummel, also in Geneva, who says, well, we should move away, of course, from one size fits all medicine to stratification. And the stratification can be done by neurophysiological markers like MEPs, for example, it's all a matter also of cost. To do these assessments in the periphery is a bit difficult. So we were thinking of how to overcome this, and I'll come to this point, how we can maybe overcome this problem. So if you look into these results, you would say, well, if you have patients who have no finger movements left, so the physiotherapist can't do anything and the patient can't ever do any training, the first step could be to use the computer brain machine interface as I've shown before with a hand exoskeleton or gait exoskeleton. And once this EMG activity is restored, you can use the EMG activity for functional electric stimulation or for triggering other tools for actuation. And once there is really some function restored, you can move on to constraint induced movement therapy. So that would be the pipeline that could be implemented for stroke patients who are severely affected. Now let's come to some first conclusions. I've shown that these assistive and also restorative BCIs or brain-machine interfaces are effective clinical tools already now to improve quality of life in paralysis. And I've shown that repeated use of this BCI technology can lead to functional and structural plasticity of the brain and spinal cord and trigger neurorestoration. The variability in the clinical efficacy is rather high. So instead of a one-fits-all approach, stratification and personalization will be key in the future. Now, the question is, where are we now with the BCI technology? Is it now ready for neurorehabilitation? And I've been working on neurorehabilitation for the last 10 years to implement BCI in stroke, and still we haven't really managed to treat all the patients at the Charité with this technique, but I'm working on it. So I'm quite positive that in the next 5 years we are going to move there, and I'll show you how. Now, the problem that I mentioned is that proper stratification may be very cost intensive, and to do all the diagnostics might not be available in the periphery. So here in Berlin, we have everything, but if you move to Brandenburg, only 20-40 kilometers from here, they don't have these tools. So, and usually these rehabilitation hospitals are outside in the periphery. So they really can't do this stratification. And now, another point is that this BCI-related sensory feedback, if it does not really have any functional meaning. So the patient is opening the hand, closing the hand, but there is no meaning to it. This may not be sufficient and sufficiently motivating for the patient to repeat this over and over again for a whole month or even several months. So that's a big problem. So my suggestion is here to combine the exoskeletons in physiotherapy and use it as a technical aid in physiotherapy for practice of activities of daily life at home or in a context that is very similar to what the patient would actually face when the patient is discharged home. So here you see an example of a patient who suffered a stroke more than 20 years ago. She is severely paralyzed and has high grades of spasticity. And we used our neural exoskeleton with a device from Sunosaki in Hong Kong, a handy rehab. And this device can help her to grasp this bottle. And with this, she can do bilateral activities of daily life, like opening a bottle. Or here is an example of opening a box of potatoes or a bag of potatoes. So for these patients, it's really the biggest challenge to make them use of both hands. So usually they learn to compensate, to do everything with the unaffected side, and that actually leads to more loss of function on the affected side, because they have this learned non-use. And now if we equip them with an assistive device that is intuitive, driven by ipsilesional brain activity, they can restore these bilateral activities of daily life. Now we have also used more sophisticated devices. So the Sunosaki hand can do simple grasping. Here you see a patient with multiple sclerosis on the left side who cannot move his right hand. And he is able, with this device, it's from HKK Bionics in Germany, to light up some matches and he's quite good in controlling this exoskeleton. And here's another patient with a complete avulsion of the plexus brachialis. So it's a flaccid paralysis, and you can see that with the device, he can move these different cylinders on a desk. So this is the more advanced version, and you can see how he's doing that. Now, there are also other devices coming on the market now. There's a lot of dynamic now, especially in Asia, but also in Germany, and of course in the US also. You have the Myomo. There is the Neomano from Neofect, South Korea. So they are developing some device, but the problem here is to overcome spasticity, and that's mainly that's the main constraint in most of these devices to address the issue of spasticity. So the handy rehab can do this to a certain degree, but well, there is still some space for improvements of the device, obviously. And the HKK Bionics device here shown on the right is rather expensive. It's about 40,000 euros. That's $50,000. But the insurance companies are covering it now. So like our state insurance, which is really a great thing now. So that is the first step to move this towards real application outside of the hospitals in the daily life of the patients, because that of course accelerates adoption if the insurances are covering the cost. Now, well, this is where we want to go. We want to go to the patient's home, and here you see the patient you've seen before with a 3D printed exoskeleton. But these devices, of course, have to be certified. That's quite a long way, and it's hard to find investors for this kind of businesses. And another issue that we have tried to address, and I think we are on a good way, is that these patients cannot use these caps, these electrode caps. Usually you need two hands, or actually you need someone else to put on these electrode caps to record from your brain. So in the last years, we have worked on headsets that are mountable by the patients themselves. So for our device, you only need 4 or 5 electrodes. So essentially, the device that you see here on the upper right is overpowered. You just need 5 electrodes for hand movements. So in the future, we will have much slimmer devices, and you will see that these devices can be put on by the patients themselves. I had some faith in these dry electrodes and the newer generation that are more flexible, but the signal quality is still a big issue. So right now, I'm more betting towards textile electrodes, so sponge electrodes that can be used for this purpose, because it's very nice to wear. Actually, it has a very high wearing comfort. So here you see now all the technical challenges, but I've mentioned we're on the way to really overcome these technical challenges. But once these challenges have been addressed, there are all the structural challenges in the institutions. So, for example, reimbursement. So I've mentioned there is now a device that can be reimbursed, so that will boost the scaling up. But also the integration treatment protocols. Right now, there's no real standardized protocol to introduce exoskeletons in physiotherapy or in any form of like long-term treatment plan. So, and also the therapists have to be trained, and the whole device has to be implemented in the pipeline in the rehabilitation hospitals. And this, of course, depends on the preparedness of the hospitals to early adopt. And here in Germany, we have now a number of hospitals who are these early adopters. So and they help the companies also to improve their devices. So I hope in the next years we have all this in place and overcome all these challenges. Now, if we introduce these devices into everyday life environments, obviously there are a number also of neuroethical challenges that we have to address. People have to be much more literate about this technology, and also we have to address issues of like data security, and these companies are using the cloud, so we need to make sure that proper encryption is used and all this data security is implemented. Now, that's more or less what I wanted to say, and I'm going to summarize now what I've been pointing to. So I've tried to convince you that these assistive and restorative brain-computer interfaces are effective clinical tools now, and they can restore movements of a variety of patients with MS, with plexus evulsions, with stroke, with spinal cord injuries. And there are many novel tools in the pipeline, but the clinical translation still remains a challenge. Using these neuroexoskeletons as a technical aid for physiotherapists may be right now the best angle to broaden the use of brain-computer interfaces. And I would say that the BCI technology is ready, but we still depend on institutional support, early adopters, so to speak. So with this, I thank you for your attention, and I would like to also mention these two special issues that are now, I'm also co-editing, so an IJNS on brain neuroassistive technology. So if you have some work that you want to be seen, because this journal is seen in the health sciences and also engineering sciences, submit it. And there is a new research topic in Frontiers in Neurology that I'm also co-editing on transitional and long-term continuous care. So this is really now trying to address how can we make sure that patients are getting a personalized treatment plan for their individual needs. So this special issue is going to go into this direction. So if you have something, please submit it to this research topic. So with this, I would like to thank you very much for your attention. I'm looking forward to the discussion.
Wonderful. Thank you, Surjo, for the presentation. I'll open the floor up for questions. Dr. Cohen, you're muted if you're trying to speak.
Correct, I am.
Okay.
Very nice presentation, Surjo. Thank you. It's a pleasure to see you again. I was wondering, what are your thoughts about the best controllers for all these novel devices at this point? And I'm referring particularly invasive versus non-invasive, and if non-invasive, single choices, like for example, EMG or eye movements, or combination of them.
Well, that's of course a very good question. Now, I have also shown that you can trigger this plasticity using EMG if there is EMG associated with a similar function. For example, in gait, when you have a clear instruction, okay, now your goal is to move your feet, and they try to do this like moving forward, trying to move their feet. And if you have some correlate of from wherever you have it, like whether it's from the trunk or from EMG activity or from the brain, it in the end doesn't really make a lot of difference if you stimulate the right afferent connections and cells. So essentially, the question of the right controller is, very complex because it depends on signal quality. So, for example, if there is a clear intention to perform a movement, but there is a lot of variability in the signal, and we have this problem with EEG because it's a non-stationary signal. Of course, the contingency is not perfectly high. So you always have much higher contingency if you have an EMG signal. But the problem, of course, if you have paralysis in the fingers and you want to move just only the fingers, the problem is that you can't really get the signal from the EMG. And that was why I suggested then, if it's really about only moving the fingers without having a complex movement associated, and it gets really tricky there, then the best approach is to use EEG activity. And of course, if you use ECOG and you are much closer, you can do single fingers even, like the index or the pinky. And we are trying to do this now with these quantum sensors. because I'm shying back a bit to implant ECOG just to do this kind of operant conditioning of these cell assemblies that hopefully in the future, if the rehabilitation works, you don't need anymore because you have restoration of the function. So that's the reason why I'm trying to make this point of increasing the spatial resolution of these control signals from the brain using maybe these OPMs in the future. Right now, it doesn't work yet, but I think in the next 3 years, we'll get there. But if you have other signals, and if it's, for example, about restoration of gait, then I think what they've done in Lausanne is just the right thing. So the controller would be an EMG activity.
Duran, Dominique.
Yeah, thanks. Very nice seminar. I got a question about the EEG itself. I'm just wondering how reliable it is for a day-to-day activity with a patient moving around. And also I'm wondering to what extent you can push it. So if you only have 4 electrodes, how many bits of information can you extract from that?
Yes, so essentially what you can do is hand closing. And this is when we compared to, like we had those patients who had plexus evulsion and they didn't have any EMG to record from. So they had to relearn to use another muscle to control, for example, an exoskeleton. And when we asked them, okay, what do you like more? Do you like the EEG control or the EMG control more? In those cases where they didn't actually have EMG in the shoulder or on the arm, they clearly said they like the EEG more because it's so intuitive. They just think about closing their hand and then the hand closes. But that's essentially all you can do. You have a binary signal. And the reason why we have included EOG, electrooculography, is that it's a very reliable signal. And just to make it safe in everyday life, we use EEG to close the hand, to have a secure grasp, and then manipulate, do whatever you need to do in the kitchen. And when you're done, you're using a horizontal oculoversion that you usually don't do in daily life. It's like a maximum movement of your eyes that you can record from actually the EEG electrodes. So you don't actually need to place more electrodes on the surface of your face or somewhere. You can record these signals and classify it from the scalp electrodes. So you just need five electrodes placed over the motor cortex, and that's it. And with this, you can do the open and close movements. But well, this, for example, if you don't have finger movements, but you have, for example, elbow movements and shoulder movements, and you can restore the function of your fingers. Of course, if you use it on a daily basis, you will improve the function of the whole arm after a while. And that's really what we want to achieve here. It's open close. So that's what we can do right now.
Thank you.
So, Surjo, maybe I can ask you a question as well. So I was wondering, can you speak to the appetite in Europe for invasive technologies? You know, I've gotten the impression that maybe there's not as much interest, in invasive, either brain-machine interfaces or even FES. Is that a true? Is that a fair statement, or how would you describe the environment in Europe? How they view invasive versus non-invasive?
Well, there is some invasive work, but I would say, well, there were so many constraints now to go into this direction. You've heard about Nikos Logothetis and Göttingen, the Primate Center. They all had problems to really push this kind of research forward. And then the translation to humans would be, of course, the next step. And I would say that a number of neurosurgery departments are very open to go into this direction. But what they want to see is a clear benefit for the patients. And that has been shown now with this work from Lausanne. So now they are very excited to implant the spinal cord stimulators. And I would say, look, in those patients, if we understand who of those patients will benefit from non-invasive treatment, and who will not, because there are obviously also people who will never benefit from non-invasive BCI training because they don't have the well, the damage is just too big to extend it. Then of course those patients, if you can show that those patients would benefit from an implantation, and you can actually also get reimbursement from insurances, you are all set. People would actually implement it here in Germany for sure. Yeah, but right now it's not so clear what the benefit for the patients is, and then they are rather hesitant to offer that.
Is it challenging to get, so in Europe is what the CE mark, is that what it is? Or,
yes, but it's very similar to the FDA certification. So it's just another thing that you have to get in Germany to actually be allowed to implant it, the CE mark. So there is Cortec in Freiburg, so they have the certification, and of course they try to make a good case for the implantation of ECOG grids. But you know, I think really in a few years, if we understand better who will benefit, and we can really predict on a very reliable basis, well, it's just a matter of a few years, I would say. And the technology advances a little bit more into these really wireless devices, and you have more actuators that you can really use in your daily life. So you have an implanted device, and you say, okay, for this patient, you have this and this device, it costs 50,000. But he can cook in his kitchen and could do everything. Then I think everyone wants to have it.
How easy is it to do like large scale clinical trials? And I ask because I was listening to a podcast recently, and they said one of the challenges in cancer research, for the longest time, is that there is no sharing of data really across institution, across PIs. And actually, I'm seeing things very similar in terms of the BCI work in the States. It's very hard to do large scale clinical trials. There's very little sharing of data. Everybody's technology is slightly different, maybe implants in a different area. And so large scale clinical trials are challenging. Are there efforts or initiatives in Europe overall to maybe sort of address some of these concerns so that some of these there can be a faster path forward for technology adoption?
Yes, absolutely. I mean, right now in Germany, but also in other countries in Europe, there is this big wave of open science has arrived here, and really the institutions are pushing that you are publishing your data along with all the results. And this is also the standard now in my lab. So I'm not publishing anything without the data and the script. So that's now our new standard. And the Berlin BCI group that was initiated by Klaus-Robert Müller and Benjamin Blankertz, they have already started to have a database of BCI data sets. But what has to come next is now, when we go into this clinical translation, exactly what you are suggesting, that we are putting some standards in place, and then also have a common data base where everyone can actually have access to. So I absolutely agree. And there is this initiative, the Clinical Brain-Machine Interface Society, that was initially founded to really to implement this. And I hope that in the next years, we will make more steps towards this direction.
Thank you. Other questions?
Maybe just a comment. I really like the data sharing approach that you've taken. I think it's way overdue. And as Baloo said, in the brain-computer interface world, it's been really pretty poor. There are problems with it. People do things differently, et cetera, to try to do comparisons. But if you want to push this into the clinic, it sort of means commercialization. So if all of your raw information is out there, will anybody ever commercialize this? Has that issue arisen? Like anybody, you know, you've given away all the information basically.
You mean your IP? Like if you've given away all your IP?
Well, yeah, basically. I mean, you know,
You're supposed to file a patent before you publish, Bob.
yeah, I know. You know, let's say, anyway, my main comment was I like what you've done, and it's been hard to get people to go along with that, I would say. Although the National Institutes of Health are really starting to crack down on this. You know, they're paying for it. The government is paying for this work, and they own it, right? So,
Yes.
anyway.
I do have another question. This is Dominic again. So the idea of this non-invasive is extremely appealing, obviously, but I don't see a path towards more complexity. In other words, how do we control a hand that has 22 degrees of freedom eventually? I mean, okay, we don't go that far, but how do you go from 1 degree of freedom to 3, 4?
Yes. Now, if you look into the work of Louis Benabit in Grenoble, he has shown that when using his device, after I think it was 5 months, the patient could control 8 degrees of freedom. But that was, of course, based on a lot of training. So you can gradually increase the degrees of freedom, but it's very cumbersome if you are away from the cells. Now, what gives me hope? I mean, of course, you can go this way and you can do this stepwise. It's a very long process then to increase the degrees of freedom with non-invasive technology, but it is possible because we can condition these different. I mean, it's all about the spatial resolution. And what gives me hope is that we have now technologies that are not available yet. But if you read, there are polymer-based quantum sensors, nitrogen vacancy magnetometers. They're all now in the pipeline, and they will really reach much higher spatial resolution than EEG or the OPM sensors that we have now. And it will just take a few years to put them out, and then of course we will have higher resolution. But until then, we have to implant. There is no other way around it. If you want to have a fast success, of course, the implantation is the only possibility.
So you believe in those magnetometers, the new ones, surface room temperature ones? I've seen them, but I'm not sure how they can be really used. They're still bulky. I guess you're right. In a few years, they're going to be
2 years. Yeah.
2 years, you think?
No. It will take much more than 2 years.
Oh, okay, that's what I thought. Yeah.
Invested into this now, I think 2 years since I've been coming to Berlin. So we had an MEG center in Tübingen that I used. It was a 275 sensor MEG, and here at the Charité they don't have an MEG, so I was actually thrown into cold water because I was using the MEG for many years, and then I was looking for alternatives, and then I thought, oh well, these OPMs are really exciting. So there's Matt Brooks in Nottingham, and he's doing really amazing work with these sensors. So we collaborate on that, and the PTB has the best shielded room, and that is already the point. So if you have a very good shield, like a very good shielded room, of course you get good signal quality. But as soon as you're moving out of these shielded rooms, you can't get any reasonable signals. But in the future, I mean, these are magnetometers. You can build gradiometers with these sensors, and also these NV magnetometers, nitrogen vacancy magnetometers, can be built as gradiometers. And that's just a matter of time that you can then use them outside of a shielded room. But there's a lot of physics that has to be implemented. And I would say it will be 10 years. Will be 10 years from now. So the OPMs will work in like 5 years in a shielded environment. And if you want to go out of this environment, it will take 10 years from now.
Yeah, I get it. I think Don has a question.
Don, do you want to ask your question or do you want me to read it?
I'll go ahead and ask. So you showed some really great, levels of improvement with the BCI driven technology. I'm just wondering how much these studies have control groups where people are simply cued to attempt to make the movement at this time, and then the device responds, so that, you know, they're still attempting to make the movement and the device is still responding. How much is the benefit of the EEG versus just timing it appropriately?
Yes, that's a very good question. And in fact, if you look into the literature and to the meta-analyses, there are some studies that have proper control, not so many. So a lot of studies, they don't really have a good control condition, but there are some studies, and they show that if you use non-contingent feedback or non-brain related feedback, it didn't really induce, it didn't trigger these neuroplastic changes. So, but obviously this is something that has to be investigated in the future. The studies, there are now 300 patients, I think, published in these controlled BCI stroke studies. So the data is still little limited, but I think in the future we'll know much more. And the problem with having a proper control is obviously that if you tell the people, okay, try to move your fingers, they will try it and they will activate the neural circuits that are necessary to do that. And if you, then you have to control for the intensity and the contingency. So, and that makes it not clear. Normally you have a control group where you have a clear separation. But here it's difficult because you ask the patients, okay, try to move your fingers. And of course they are going to activate something, and that makes it quite difficult. But the studies that have been done and properly controlled with fake feedback, brain unrelated feedback, they clearly show that the contingency of the EEG signal has an impact.
Thanks.
Okay. If there are no more questions, then please help me in thanking our speaker, Dr. Surjo Soekadar. Thank you, Surjo, very much for joining us. It's a shame that, you know, we can't bring you here now, but, you know, I look forward to the next time that we meet.
And working late in the evening. Thank you.
And working late in the evening, yes. Yes, thank you.
Best time.
Thank you all for coming. The next MP seminar is when?
Well, right now it's May. There'll be others though that come up, but yeah.
So please keep a lookout.
Yeah.
Okay.
Thank you very much, Surjo.
Thanks for inviting me.
Thank you.
Bye,
all.
Leo.